Hello, welcome to Science Hangout from Science Mission. So we have today Dr. Cheryl Rosenfeld from University of Missouri with us. She is a member of various professional associations. She also is the reviewer for various journals. Dr. Rosenfeld has been serving various NIH peer review committees for last several years. She has authored several publications, articles in very well-known journals, peer-reviewed journals. She is featured as a Scientist of the Week from Laboratory Instruments in 2013. Welcome, Dr. Rosenfeld, to Science Hangout.
Thank you. It's a pleasure to be here.
Can you please give us some background about yourself, your education, and some of your research work?
Yeah, so I am both a DVM and a PhD. I got my DVM from the University of Illinois Urbana-Champaign in 1995, and then I got my PhD degree at the University of Missouri at Columbia, and I've stayed here at University of Missouri at Columbia as a faculty member since 2004. And my research looks at how the maternal and the paternal environment can affect later offspring outcomes. This phenomenon we call development origin alpha disease, and the idea that essentially what could happen to the mom and the dad can have long-lasting consequences both from the health and from a disease standpoint to the offspring.
And what makes you motivated to work every day to come to the lab?
Well, I've always loved science, and to me it's always a mystery what you're going to find. And to me, I like the idea that hopefully my work is going to have long-lasting consequences in terms of helping promote the health of offspring. Just from my own family experiences, we've, I've seen just how much the in utero environment can shape later health outcomes.
And in your career, do you have any role models, mentors who helped you?
Yeah, I've had, so I think, to me, I've always just loved a lot of the scientists like Jane Goodall. But I was fortunate when I was in high school to have a really amazing AP biology teacher. I think she really, focused my studies and just my interest in science, and her name was Patricia Murphy.
Okay. And what are the challenges do you face every day?
I think the challenge is you just never know what you're going to get into when you start an experiment. You think you might have had all the confounders planned for, but that's never the case, unfortunately. I think to me, the other obstacle I think a lot of us are facing in the scientific community is just the reduced NIH money is always a concern. To me, I think people need to realize the work we do in the laboratory can have really dramatic consequences in terms of their own health. And I hope that if we start getting more monies into NIH, I think that's been a major obstacle, both for me and my colleagues.
And in your spare time, I know you are busy with your work and all, during weekends, in your spare time, how do you spend some free hours, and what are your hobbies, so to say?
So I guess if I hadn't gone into animals, I would have done plants, and so that's become my hobby. I do a lot of work in the garden. Again, I've been fortunate because living in Missouri, we have a lot of wooded acres. So I do a lot of work there, hiking, birding. So I guess and traveling as well. So tomorrow, in fact, I leave for Costa Rica.
Okay. And can you please talk about your lab people and all?
So I have a variety of lab people. Again, I've had postdocs, I've had grad students and undergrads, and I encourage all to publish. In fact, so the work we're going to be talking about today, the first author on it was she was an undergraduate student in my lab at the time, and I just thought she was a phenomenal student, and I thought, oh, I'm going to put her on this project, and it turned out all right, okay, at the end.
Yeah, that's good. And how do you motivate your students?
I think to me, the way to motivate the students really is just to take pride in small achievements. I tell them it may be something that's only of interest to you, but if you find the answer to that, that's in itself important. I mean, So it's not always going to be the nature of the science papers, but just little small things along the way that might be of interest.
Mm-hmm. Like I read one of your recent articles in scientific reports, like about the bacteria in the seminal fluid.
Mm-hmm.
So how did you come about that? Like what made you think of in those terms?
Well, we know that the microbiome in the gut can have a huge consequence in terms of various health disorders, whether it be diabetes, obesity, or neurologic disorders.
Mm-hmm.
But again, in my interest being in developmental origin health and disease, I was trying to think of how the dad can influence his offspring. And we know that their germ cells obviously can play a huge role. There was also then a report that came out in PNS I'll be showing today, which showed that the seminal fluid secretions could also impact the offspring. And I thought, well, the seminal fluid is an important environment because there's a lot of nutrients in there, just like the gut. And I thought perhaps that would be a place that the bacteria could reside in. And it would be a unique composition, though, because the nutrients within the seminal fluid are quite different from that which would be in the gut.
Okay. And only you think like only certain kind of bacteria can grow there?
I think so, because again, like it's always going to depend on what kind of nutrients. Obviously, what can you feed them basically can help in terms of. So yeah, that was one of the things we wanted to compare back to was a microbiome that was well established, and so the gut microbiome was the one that came to mind.
Okay. I think without further ado, I would like to learn more about your work. So
Yeah,
interesting. Could you please go ahead and present your work, please?
I'd be glad to.
Thank you.
So today I'm going to be talking about the discovery of a seminal fluid microbiome and how it might influence health of the male and his offspring. But before I do that, I just want to give a brief background into developmental health and disease. And again, what basically this is that the environmental condition that organisms, including humans, can experience prior to birth can dramatically shape the later health for better or worse. And this first concept was first articulated by the late Sir David Barker, and he coined it the Barker hypothesis. But to give a more idea of what it entailed, it was then changed to fetal origin of adult disease, quote, for the abbreviation. The thing about that, though, it implied that everything that happened to both, let's say, the mom or the dad would have negative consequences to the offspring, and we now know that may not be the case. So more recently, it's been changed to the idea of development, origin, health, and disease, DOHAD. And this idea has gained increasing currency because what it may do is explain many what we call non-communicable diseases. These are diseases that are not transferable across organisms, but can play a great role in many organ systems. And one thing I would like to point out is when this idea was originally coined, it essentially was that the idea was more so that the mom would play a role. And again, in this book, it kind of illustrates from the 1970s Mommyism, the silent disease of America. But we now know that it's not just moms that can play a role, it's also dads that can play an important role in terms of the health of their offspring. And on this aspect, there was a paper that came out in 2014 that looked at the maternal tract factors that contribute to the paternal seminal fluid and that could have an impact on the metabolic health of the offspring. Essentially, what they did in this paper was that they removed the seminal vesicle, these accessory glands called the seminal vesicles, and they could see that these offspring that were born from these males they had outwardly looked normal, but they had severe metabolic phenotypes. And so just basically by taking out the accessory gland, they could then have later consequences on the resulting offspring. So if we look here, in terms of when could mom and dad shape the later health of the offspring, there could be several periods. First, we could have during the periconception, which would be prior to fertilization. So we could have disruptions in the oocyte maturation. That would be on the mom's side. And the dad's side, their germ cells are called spermatozoa, and they could have epigenetic or phenotypic disruptions. The second time period that we can have problems is the periconception prior to fertilization. This is where we could have small RNA or transfer RNA changes in spermatozoa that may occur during spermatogenesis, i.e. during the development of the germ cells, or as they have to go through this tubulus called the epididymis. And this came out actually in Science just a couple months ago, showing that these transfer RNAs could also play a role. But as I showed you in that one paper, the seminal fluid secretions can also impact zygotic development, and it can interact with female reproductive system to do so. So this is where, again, I'm coming in from this point is the seminal fluid composition. But just to look at some other things that may affect the later health of the offspring, that could be during the in utero period. That would be in the case of mom. You could change environmental factors, like, again, expose them to environmental chemicals or a high-fat diet or obesity. And again, this could both affect the placenta and the fetal development. There also could be consequences during the postnatal period, such as alteration in milk composition, the transfer of environmental chemicals or stress hormones. There may be insufficient colostrum transport. And again, the colostrum is what protects the neonate against some infectious diseases. Now, during the postnatal period, we also could have compromised parental care, and this can be both on the mom side and the dad side in the case of species that are monogamous and biparental. But what I want to come back to is the seminal fluid. And this, after reading that paper, it got me thinking that there could be a microbiome that lives within the seminal fluid that could be transferred to his reproductive partner and then affect offspring outcomes. So I want to illustrate to you what these seminal vesicles are. So this would be a male, and what you can see here, they are a paired structure. They almost look like a uterus, so we sometimes actually call them a horn structure. But here's one seminal vesicle, and this is another seminal vesicle. And if we look then at the under the microscope as to what the seminal vesicles will look like, they're essentially these large alveolar glands, and you can actually see the secretion right there. And the secretion contains a lot of carbohydrates, especially fructose, but there's also a lot of other nutrients that could serve as a substrate for the bacteria, and this could spare the bacteria could presumably reside in this area. Now, right about this time, we started to do our work in terms of trying to isolate this microbiome from the seminal fluid. And it was quite interesting to us because at this time then, in their Cell paper, Rando and Simmons speculated that there might be paternal transfer of microbiota to mates or to offspring. So essentially they had the same idea, but hopefully we had a bit of a head start in terms of trying to discern this microbiome. Again, prior studies have identified select bacteria in men's semen. The problem with these studies, though, is it's not clear the origin of the bacteria because they could originate from anywhere in the urogenital system. So again, we sought to determine if a resident microbiome population is present in seminal vesicles that could influence the health of the male, his female reproductive partner, offspring, and even by transgenerational transmission, his descendants. So to do this, we basically took fecal samples as our control because we wanted to compare it back to the gut microbiome and the seminal fluid directly from the seminal vesicles. So what we did was, this was under sterile conditions to minimize any bacterial contamination, and we just oozed the seminal secretions into our tube, and the bacteria then DNA were isolated and subject to 16S RNA sequencing. I should point out the kit we used, it removes the host cells, so those are not a factor, and they would not get amplified with 16S RNA in any case. It also removes the dead bacteria. And I'm going to prove in just a bit that these bacteria truly are alive. And then we did bioinformatics and amplicon analysis were performed to look at the different bacteria, or also we call operational taxonomy units, that we identified. To illustrate there, this is our bar plot of the bacteria classes between the fecal and the seminal fluid samples. And hopefully you can see when looking at these operational taxonomy units, these different genera, that the fecal samples are quite different from the seminal fluid just by looking at this. But we also did that we call PCOA to compare the fecal and to the seminal fluid samples. And by using Manova, as you can see, they were highly different from what you would see in terms of the fecal versus the seminal fluid.
Mm-hmm.
So next, we also then looked at a cladogram to see which were in terms of which genera you were unique for the fecal versus the seminal fluid. And what you can see, those that are in green characterize the seminal fluid. Those are red, are characteristic of the fecal samples. And there's huge differences in our cladogram analysis. When we move to a Venn diagram comparison, while there is some overlap of genera between the seminal fluid and the fecal, for the most part, what you can see, they are quite different in terms of the genera between the seminal fluid and the fecal samples. Now, what we then next did was we compared the classes using this program called LEFC. And you can see those in green are characteristic of the seminal fluid, and those in red are characteristic of the fecal. And this also shows you doing a p-value comparison. So this is to show that there truly are statistically different. And these would be the bacterial classes that are for the seminal fluid and the fecal samples. We then wanted to compare, though, the wild type to what we call the estrogen receptor knockout. The reason we chose these estrogen receptor 1 knockout mice is it was shown that this gene can play a huge role in terms of the seminal vesicles. In the estrogen receptor 1 knockouts, they actually have huge seminal vesicles. And presumably that's due to increased fluid secretion, which could alter the bacterial composition within this organ. So that's why we chose the estrogen receptor 1 knockout as to characterize the seminal fluid microbiome. And what you can see in our Venn diagram is there is a considerable amount of overlap between the wild type and the ESR1 knockout, but yet there also truly are differences between them. Okay. We next then used a metagenome-seq program where we could then look at which genera actually are different between the wild type and the estrogen receptor 1 knockouts. Those that were greater in the estrogen receptor 1 knockouts, and I apologize, it's for all small text, but I'm going to illustrate one in particular. These in red are greater in estrogen receptor 1 knockout. Those in blue are greater in the wild types. And I want to draw the attention to this one bacteria called P. acnes. I'm going to abbreviate that way. And it was greater in the wild type than in the estrogen receptor knockout. And this is an important bacteria that I'll be talking about in just a bit. Next, we then want to look at see how if these bacteria are changing, how does that potentially affect the metabolites? Because we know these bacteria can then convert the nutrient substrates into different metabolic products. And we're using a couple of different programs. We used a metagenome-seq with the pie crust, and then I had an R code that we got from a group at Baylor University, so we could actually look at how each individual genera affected the different metabolites. And if you notice here, one of these in particular that we are looking at is this P. acnes. And as you notice, if you see this oxidative phosphorylation, this box in red means as this bacteria goes up, so too does oxidative phosphorylation. Those that are in blue, you can see some blue boxes here as well, that means it decreases with the bacteria. So if you got more of this bacteria, that would mean less metabolic product. But this is one that was increased with P. acnes. Now, one of the things we then next want to do is we want to show that these bacteria truly are alive and actually could grow on a plate. So we cultured the seminal fluid under 3 types of conditions. We did a regular blood agar, we did a pre-reduced anaerobic blood agar because that's how P. acnes would grow, and we did a McConkey's for the gram negatives. So the 2 blood agars would generally get the gram positives and the McConkey's the gram negatives. So this here is our pre-reduced anaerobic blood agar plate, and what you can see here is that you've got these colonies coming along that would be suggestive of P. acnes. We then took these colonies and we gram stained them, and they are gram-positive bacilli, further suggestive that they are indeed P. acnes. But we now have a new system we can use where essentially we can pick one of these colonies and then put it into our multi-sequencing, and then it will type it based on the database. And what you can find here is that when we picked one of these colonies, these were the 4 top choices that were given by this Bruker technology, and indeed it shows that it truly is P. acnes. So this demonstrates that these bacteria that are residing within the seminal fluid are alive and we can culture them. So, in conclusions, the seminal vesicles indeed harbor a unique microbiome. The bacteria residing with this organ may influence the reproductive status of the male and his overall health. And I do want to point out this P. acnes is more abundant in the wild type than in estrogen receptor knockout. And the reason this bacteria is important is because in humans and in rodents, this bacteria is a causative agent of chronic prostatitis that can culminate in prostate cancer. And in 2012, this prior paper demonstrated that wild type mice that are in a certain background called a TRAMP background exhibit a greater incidence of prostate cancer than ESR1 knockouts. And what we believe is it might be due to the increase in this P. acnes bacteria in the wild type compared to the estrogen receptor knockout that are leading to this change in the risk of the prostate cancer. The other thing is with P. acnes is correlated in the wild type mice with increased oxidative phosphorylation that can result in damaging free radicals. And lastly, the seminal fluid microbiome changes may affect the female reproductive partners, offspring, and potentially future descendants. And right now we are working on this arm of the pathway trying to see how that seminal fluid microbiome might affect his offspring. And with that, these were again some of my collaborators. Some of them who participated. We've also been working with the bacteriology lab in terms of doing the cultures. But I'm grateful for all our funding sources as well that help support this project.
Great, nice presentation. Thank you very much. It's a pretty good presentation, and you unequivocally shown that these bacteria are alive in the seminal fluid. So I have a few questions, if you don't mind.
No, that's fine.
One of the things is like how it is like you have seen it in mice, right? Have you tried anything with the humans, like, because especially with those bacteria, it could be a biomarker if you see them early on?
Definitely.
But,
we have not. You said like they have done semen samples from men, but the problem is again like where do the bacteria originate from? That is a true concern. So I have not yet moved to men, but what we are trying now is essentially a couple approaches. I would like to see if we put antibiotics into the seminal vesicles and try to destroy off that microbiome, then breed those males, how do his offspring turn out? I think to me that would address this idea of development, origin, health, and disease. We are also then, another approach we're doing is we're trying to get those colonies that we've isolated, and we're going to put that back into the female reproductive tract and see how it influences her health as well.
Yeah, like, and what are the what do you expect the female would have? Like, what are the kind of like diseases they might develop because of men having this kind of bacteria in them?
Well, again, the question is whether it's actually this could be a potential cause of the innate infertility. The other thing is, again, it could affect her menstrual cycles. Cervical mucus secretion. So there's several ways whereby the seminal fluid microbiome could influence our health. The other thing I would suggest is that what could also happen is that the bacteria living in the seminal fluid microbiome may take up residence within the vagina and help to establish a population there as well. So again, there could be also long-term consequences to these transferring the seminal fluid microbiome.
That's interesting. I mean, like there is a lot to be done with your recent finding, I would think.
There's a lot of different ways we can go with it. But I think from my perspective, I'm more interested, again, like I said at the outset, about how he can affect his resulting offspring. I think that we do need to think about how the seminal fluid can affect the health of the man, especially with this one bacteria.
So, like, you think it is kind of like you can transmit this bacteria from the male to the women and from there to the kids, or like whatever the effect it might have?
Well, and I think
bacteria.
Yeah, and I think the thing that you have to think about is that during that early embryonic period, those bacteria could still be present in the female reproductive system, and they could be influencing it. I mean, what amazes me as you think about it is that the bacterial, if you combine all the bacteria together, they have more genes than we do in some ways. I mean, so there's a lot of influence that could be going on. The other thing is, besides metabolites, they can actually make hormones that replicate our own hormones. They can listen to our hormonal signaling dialogue. The other thing, we didn't see so many of the gram negatives, more so the gram positives. But again, there are virulence factors like LPS that could play a role as well too. So I mean, it's not just the bacteria, but it's the bacterial metabolites, essentially all of their products that could be having effects in terms of these downstream consequences.
Do these bacteria help grow other bacteria which are kind of not usually seen, like help developing certain population of bacteria more than the others?
I don't know that. I don't know the answer to that. If that's it's the thing you'd wonder though, if they can outcompete each other more so. I mean, and one of the things we looked at was what we call the diversity. There's less diverse bacteria in the seminal fluid than the gut, but you'd expect that because it's a smaller area. But one or two populations take over, it could then outcompete the others. I mean, I think that's the biggest concern as to what are the ones. I mean, we talk about dysbiosis in the gut, and perhaps there could be a dysbiosis within the seminal vesicles as well.
Okay. Yeah, it's interesting, and it has a lot of implications to the human health.
I think so too.
Yeah, it's thank you very much for your presentation and discussion with us, and we look forward to seeing more of your publications and work in the near future. And we invite you further, like if you get something new, certainly we'll happy to share it with our audience
I would love
through
to.
you.
I'd be glad to. I hope to come back.
Thank you so much.
Okay. Have a good day.
Thanks.